definitely not available to speak. Therefore, that helps us a little bit with the schedule. Professor Ravi Bellamkonda, who will introduce in a second, has 2 talks. We will have him give the first talk, take a short break, and then have him give a second talk. Then we'll have a slightly longer break as scheduled, and then we'll go into the roundtable discussion. So it's a pleasure to introduce Professor Ravi Bellamkonda. Ravi originally from India, did his PhD at Brown University, although most of the time was spent in Switzerland, as far as I can tell. And in fact, he then started his academic, did a postdoc, started his academic career at Case Western Reserve University, and 2.5, almost 3 years ago, moved to Georgia Tech to our biomedical engineering department. He heads our neural tissue engineering effort. And Ravi, it's all yours.
Thank you very much. Just making sure this works. Does this work? No. Okay. Maybe I'll just use one of these. Can the gentleman help me with this?
Is there someone that can help with the microphone in the back of the room? U.S. delegation, after Ravi's first talk, would like to get everyone together outside this building, take a picture of all of us together.
Okay, thank you.
Now we're working. Okay.
Okay, so thank you very much. It's a pleasure to be here. I'd like to thank first Bob for including me in the delegation, and the folks at NRC and Professor Marai. I think I should say something like Shokran Le Devat Kom or something like this. I was trying to practice it, but it's not coming out quite right. But so I'm happy to talk to you. In the first of my talks, I'll talk to you about peripheral nerve regeneration. I think the order is kind of logical in the sense that it's perhaps a little bit easier to get regeneration per se in the PNS than in the CNS, and I'll tell you why as we go along. So just for those of you who are not, neurotypes, I just want to clarify the difference between PNS and CNS. The simplest way to think about it, I suppose, is that everything that's inside your cranial cavity and your spinal cord, your vertebral column, is technically CNS, and everything that's outside is your PNS. And the difference is significant, not just anatomically, but in the regenerative potential, because amongst other things, the cells that myelinate the peripheral and central nervous systems are quite different. And not only are they different, the way they respond to injury is very different. The Schwann cells in the PNS respond with a phenotype that actually is pro-growth, whereas the oligodendrocytes and the CNS myelin itself, many people believe, is very inhibitory with some of the myelin proteins in there. But we'll talk about the CNS in the second talk. On this talk, I'll concentrate on the PNS, which is outside, all of the nervous system that's outside the spinal cord and the So, why bother with PNS? I'll tell you a little bit in a couple of slides about the typical issues that we face. I'll tell you what the current clinical approach is, and then I'll discuss a couple of different approaches using tissue engineering to repair peripheral nerve gaps. And in the end, we'll try to draw some general conclusions about where to go from where we are. So generally, you get peripheral nerve defects as a result of trauma, an accident of some kind. More often, actually, it's tumor dissection that causes peripheral nerve gaps. Whenever there's a soft tissue tumors, especially in the head and neck region, and elsewhere, when you resect tumors, prostate is another example. You get, this is an example of the prostate in prostatectomy, you get peripheral nerve gaps. And also in plastic and reconstructive surgery, create nerve gaps. All right? So the current clinical approach, and I'll show you a few slides, is to use autografts, but I'll show you a couple of slides on the general progression of what happens when you injure a nerve and you resect a tumor. So this is a cartoon that I borrowed from a review that Rajiv Meeta wrote. He's in Canada, in Toronto. And essentially you have the epineurium, your perineurium, and your endoneurium, the three membranes that surround the different, you'll notice that it has a fascicular structure, your peripheral nerves. And then when you damage this nerve, typically you have degeneration, and it's called Wallerian degeneration. And the thing that to appreciate with nerve repair, which is different from perhaps bone repair, is that you're not just interested in bridging a peripheral nerve gap in the sense of having a nerve grow from point A to point B. When you resect a nerve, everything distal to the site of injury actually degenerates. All you have left is a structure with the glial cells and the sort of empty tubes or channels left behind on the other end. So for you to recover function, you have to have nerves regenerate across the gap, enter the distal end, grow all the way through the distal nerve, make functional connections, and then your brain has to relearn how to map those connections to actually control it. So the problem is quite different from bridging a bone where you're just interested in healing this fracture right here. In a nerve, after it crosses the gap, you still have to have all these other things happen. And you can appreciate that because the cell bodies are actually displaced to one end, the proximal end, and that's the situation. So after injury to the PNS, there is an attempt to regenerate, and the degree of success depends on the extent of the gap. So if the gap is very, very short, you do what is called as coaptation, where you just bring the two ends of the nerve together. This is epineural repair. So you just take the two ends of the nerve together, you kind of try to match approximately the fascicles, and you put them together and you suture them. All right? Again, if you think of this as proximal and this as distal, you just want these nerves to grow along. And remember, these channels, after you cut it like this, everything distal actually is dead. The neural tissue is dead. So the nerves that grow from here have to enter these little holes in here and then go all the way across and make neuromuscular junctions or wherever the original synapses were. But this is the most common procedure, and it works actually surprisingly well with epineurial repair. If the surgeon is really, really, interested in getting this to work, they can sometimes do intrafascicular repair, where they can take individual fascicles and try to match them up and suture the individual fascicles together, which are groups of nerves. But that's very time-consuming and not done very often. And the gap is a little bit longer, so you can't bring the two ends together. You don't want to put tension on the nerve so quickly, okay? So there is interesting research that you can slowly put tension on a nerve, like when I'm growing up and my nerves are shorter and now they're longer. So that's okay, but if you want to try to put tension to put nerve together, actually that's one way to kill the nerve. So when there's a short gap, actually you do get regeneration, and one of the most common strategies used is a nerve guidance channel, where if you have a channel like this, and these are made out of polymers or biopolymers like collagen and such, and you get essentially a fibrin cable that forms, it tapers from the end, and then Schwann cells invade the channel and align along the nerve, all right? And then you have nerves that follow and the nerve gets bridged. So in animal models, in rats usually, about 5 millimeters or so, you can get very good regeneration using just an empty channel. There are differences of whether you use a semi-permeable channel or a non-permeable channel. Initially, people started working with silicone non-permeable channels, and then we realized that a semi-permeable channel with about 50,000 molecular weight cutoff was probably better. The series of papers optimizing channel properties. But essentially, if you increase the gap to about 10 millimeters, then putting a channel doesn't work anymore. All right, so it has to be only 5 millimeters or smaller, and mainly the conventional, the dominant hypothesis is that this fibrin cable that forms from two ends is unable to form very efficiently as you start increasing the nerve gap. And therefore, there is no substrate for nerves to grow on to actually make this connection. So the other thing to appreciate about neurons and nerves are that they're anchorage dependent. So if you're culturing neurons and they're floating, they're probably dead. So they're always anchorage dependent. There are no floating nerves really in your body. They always grow on something. The other thing to appreciate is I see as a reviewer on study section sometimes even to this day, people trying to modify the surface of these tubes inside, which still baffles me because the nerve never actually touches the inside of this channel. When you make such a channel and you want the nerve to go across, there is always the bridge that forms and the nerve goes in the middle. So the attempts to modify the internal surface of these polymer guidance channels, unless they affect this process in some way, are really not a good way to go in my opinion. So, but this is what happens when you use nerve guidance channels. But as I said, when the gaps are longer, just to put things in context, the human gaps that we wish to bridge are 40 millimeters, 4 centimeters or longer, 40, 80 millimeters. So we're not talking about 5 millimeter gaps in humans. And that's the problem. Okay. So when the gaps are longer, clinicians use autographs. And you typically take your sural nerve, which is at the back of your leg, and it's a smaller nerve, it's mostly sensory. The other thing to appreciate with peripheral nerves are most peripheral nerves are mixed nerves. They have motor and sensory components in them. Your sural nerve is mostly sensory, and you typically use, it's a small nerve, so you typically use multiple segments to bridge a reasonable sized nerve, and you use autographs. And this is really the gold standard, and actually it works reasonably well, but there are issues. And the sural nerve exists for a reason, and so a second surgery is not fun to do when a patient is already sick. As I said, you need multiple nerve grafts, and there are limited availability of autologous nerves. Sometimes there's just not enough sural nerve. And even in the best case scenarios, when you use things, if you do studies, and there's a nice review paper on this, looking at clinical outcomes and behavioral recovery, you only get recovery in about 40 to 50% of the cases, and this gets worse with the gap getting longer, obviously. There's also now a growing appreciation. The recent papers, there's one by Nichols, showing that the modality of the autograft that you use matters. So to bridge a mixed nerve, which most of our nerves are, containing sensory and motor, if you use a purely sensory autograft, which is our most common autograft, like the sural nerve, it doesn't work as well as using a mixed nerve autograft. And there aren't mixed nerve donor sites available that we can really use. So we're beginning to appreciate that the modality of the autograft may actually matter in the outcomes that you get. And that might explain some of this lower percentage recovery. So clearly, there is a need to come up with a better way to do this, and the question then is, as a tissue engineer and biomedical engineer or a biomedical scientist, is there anything that we can learn both from our understanding of how autografts work when they do work, and from our understanding of developmental biology and how the fetus wires us up? How is it that in a baby, the cell bodies, the scale of this is really incredible. You have a 10-micron cell body in your dorsal root ganglion or someplace like that, just outside your spinal cord, that sends out this long process that innervates, the tip of your finger. And I know I've touched it because this nerve ending is here. So how does that happen, and what are the principles that the fetus uses to guide growth cones, the tips of growing nerves, to make connections, and can we borrow some strategies from there to see if we can improve the situation? So those are your two sources of information to design or engineer a construct. And I'll tell you a little bit about how we might go about doing that. So where do we start? As I just said, in developmental biology, if you look at what are the strategies typically that nerves use to find their appropriate targets. And there are three kind of principal ones, and there's some others, but they fall under these three categories. One is haptotaxis, or differential adhesion. It turns out that in developmental biology, when nerves are just beginning to grow, pioneering growth cones, if you will, have a sort of paved and non-paved road. It's like they have extracellular matrix consisting of proteins and glycoproteins that they like, bordered by usually chondroitin sulfide proteoglycans, which they don't like. And so they have a pathway which is permissive and stimulatory to growth, and then surrounded by edges which are non-permissive to growth. And so along this road they go, if they wander off and hit the non-permissive thing, they come back and they hit the other thing. So there is a haplotaxis, a differential adhesion and repulsion working together to make this pathway. So like having a nice highway versus not a nice highway. The other thing is when they come closer to the target, usually there are chemoattractive, or now we also know from the netrin family, chemorepulsive proteins that your target cells or your non-target cells can produce. And it turns out that growth cones can sense gradients of diffusible trophic factors. Now these things usually are immobilized proteins, and these are diffusible agents. And there are a whole family of neurotrophic factors, BDNF, NGF, NT3, NT5. All of these fall under the chemoattractive category, where the target emits the signal saying, hey, this is where you need to make a target. There is a variation of haplotaxis also where you can have a cell on the surface. You can have a so-called guidepost cell where the nerve can touch a cell and it knows it's on the right way or it's not on the right path. The other category that developmental biology uses is this principle of contact guidance. Particularly in our cortex and in other regions, in the pioneering, the cells are born in the so-called ventricular zone, and they migrate to the marginal zone, and they migrate using this thing called radial glia, where you have glial cells that are set up in a pattern from the ventricular zone to the margin of your cortex. And cells that are born in the ventricular zone latch onto these pioneer glia, migrate up, and then latch off. And that's how we have the layers of our cortex. And in fact, you know the birthday of the cell based on how far up it is on the cortex. The older cells go off first, the younger ones have to go further and then hop off, and the younger ones go further and hop off. So they use these radial glia, which are like filaments that are about 1 micron scale or a little bit smaller, to latch onto and then latch off. So we know that, and also we know that several nerves follow other axons. They latch onto other fibers and then they go on. So these three principles help growth cone guidance in developmental biology. We also know from autographs that if you used antibodies to certain extracellular matrix molecules called laminin, if you take an autograph, which is the correct clinical practice as I told you, and you use an antibody to laminin, it doesn't work as well. If you remove the cells, the Schwann cells, they don't work as well. Okay, so the Schwann cells, in fact, when you put an autograft, the signals you're getting from an autograft have to do with structural signals, which is empty channels, because you've taken this autograft out and it's a dead nerve. You're just using it as a bridge, except for the glial cells that are present in there, which are alive. And so in an autograft, you have extracellular matrix molecules like laminin, and you have some structural cues. And then the Schwann cells actually produce nerve growth factor and BDNF also. So you have all this information, and now you can think of beginning to engineer a system based on that. So given what we know from, as I said, from developmental biology and from our understanding of how autographs work, you can think of a bridge, an artificial construct that you want to engineer and design. You have a proximal nerve end and a distal nerve end. You need to design it such that whatever you put in here supports neurite extension and nerves like to grow in. You can introduce, perhaps, if you use a polymer here, perhaps you can bind things to the backbone to introduce the so-called haplotactic component, where if you know the proteins like laminin, which are responsible for haplotaxis, perhaps you can bind them here. If you know the chemoattractive proteins, perhaps you can embed within this, microparticles like Pat Staten was telling us about, or in our case, we do something else. But you have some system to release a trophic factor that attracts the nerve to come in. But then you need to time this such that it's not so nice here that the nerve just is happy to be here and doesn't want to go on. In fact, that happens sometimes. It's called the so-called growth factor oasis, where if you make this too nice, then the nerves just come in here and just want to stay here and they're happy, they don't want to go on. So you have to time that. And then you may introduce some structural elements. Perhaps you can put some things up here that go this way that nerves can latch onto and grow. So these are ways to implement the sort of strategies that the fetus uses to regenerate nerves. In the CNS, if you had a similar gap, and this is all true for the peripheral nervous system, in the CNS, this doesn't work necessarily. The principal problem, and I'll talk to you about this in my next lecture, you get also scar tissue forming. And we've heard a lot about scar from Steve Badlack. Actually, there are lots of interesting parallels. You have scar tissue forming typically at the distal end, and even if you make this friendly, they don't get around the scar in the CNS. But in the PNS, fortunately, we do not see that. And if you get them to grow, and if the conditions are right, they do go on to make their distal connections. So I call this, in the PNS and CNS, a story of two things, bridges and dams. In the PNS, the story and the issues really have to do with the design of the bridges, these things. In the CNS, the story really has to do with how do you get around the scar dam which stops stuff, okay? So that's a good way, in my mind, to think about the PNS and CNS and the problems associated with regeneration. Oh, sorry, the other way. So I'll tell you two different strategies that I'll tell you actually several technologies that we use in our lab to implement those design criteria that I laid out for you earlier, the haprotactic cues, the chemotactic cues, and the contact guidance cues. And in the end, I'll tell you which way I think we should go. So the first thing was to find a three-dimensional gel that supports neurite extension. And during my graduate work, actually, we identified a subfraction of agarose, which is derived from seaweed. And this is a hydroxyethylated version of agarose. On the backbone of agarose, you can substitute hydroxyethyl chains, and depending on the length of those chains, you can control the gelling temperature of agarose. And so we use a kind of agarose that actually gels at about 17 degrees centigrade. And if you mix in the solution of agarose with no modifications, and you put, this is a dorsal root ganglion, which is a collection of neurons, they grow embedded within the gel, they send out beautiful neuronal processes. These are individual fibers growing out. All right? There are some glial cells within the dorsal root ganglia. This is the embryonic chick DRGs, E9. You do get some of these glial cells migrate out, but generally they're very supportive of neurite extension. The question now is, for this bridge that we were thinking of designing, so we have some kind of a backbone polymer that supports neurite extension. Now the fact is we want to implement our design criteria, and the first step is to implement the haprotactic element. And for this we choose laminin, laminin 1 actually. Laminin is a family of proteins. Laminin 1 is very potently stimulatory to nerves, and we decided to couple that with hydrogel. This is the structure of laminin 1. It's very large. It's about 900,000 molecular weight. We know the regions of laminin that actually stimulate neurite extension. It has an RGD segment. It has a peptide called IKVAB. It has a peptide sequence called YIGSR. There are different parts of laminin that we know already that are responsible for its neurostimulatory effect. And if you couple laminin to the backbone of agarose, we have a couple of different chemistries, and I can give you papers on those. If we covalently couple laminin to the backbone of the gel and culture dorsal root ganglia and measure the length of the processes, they grow very well in agarose, as I showed you earlier in the picture. But if you couple laminin, you can double that almost. You get a good 80% increase in the length for the same time period, so they grow much better when you couple laminin. If you couple a mock protein like albumin, so just to rule out the fact that this is imidazole chemistry, by the way, that we use in this case, you do not get that effect. If you mix the same amount of laminin that you actually covalently coupled into this gel, you do not see the effect. You have to covalently immobilize it. Mixing it doesn't do it. And the reason we think mixing it doesn't do it is because there's beautiful movies of this. The way you have your nerves extend is by pull and push. So they grab onto something and they pull themselves and then they push. And there are some beautiful movies of this. If you take a very fine film and you grow nerves on it, and if you let the film be free floating, the film actually wrinkles. It's like putting your handkerchief or a piece of paper on thing, and if you do this, the paper wrinkles if it's free. And there are movies of this proving this traction hypothesis of nerve growth. Anyway, so we think that's why you need to actually immobilize them and you can't have them floating. So the next step was to introduce the sort of chemotactic element, which is to introduce within the gel diffusible proteins that will attract the nerve to come in from the proximal end. To do that, we have actually a couple of different technologies in the lab. I'll talk to you about the first one here, and the next talk I'll tell you about some other strategies. So these are lipid microtubules. They're small, hollow cylinders made out of lipids. They're phosphatidylcholine based. They self-assemble under certain conditions. We didn't invent these. These were first invented by the US Navy to coat ships and the paint to stop barnacles from sticking to ships. We decided to adapt that to a more friendly approach. And we basically, these are the dimensions of each of these lines you see here. They're about 40 microns in length and 0.5 microns in the opening. Okay, very high aspect ratio. Think of them as hollow straws that you drink Coke with, except it will take you a long time to drink Coke with these. But so they're about 40 microns by 0.5 microns. And so when you dry these out in the presence of a sugar like trehalose, they're completely dry and empty. And you can hydrate these straws with a concentrated solution of, we've done condensed DNA, we've done trophic factors, we've done different proteins of all different kinds, including siRNA. We've done all sorts of things with this technology. But essentially, they're empty straws. You take a concentrated solution. Through the capillary effect, you can load them up. You can centrifuge them down and wash off the external stuff. And you have a slow-release system because by fixed law, you just have diffusion coming out only from the ends of the tube when you have all this solution in the middle. So you can use this system to do slow release of proteins for about 4 to 6 weeks without a problem. And we've actually published at least 2 papers on this. This is one of them. So if you make those things and put nerve growth factor in the middle of those and load them up with nerve growth factor and take a gel and make 3 layers of a gel. On one side, we put saline-filled tubes. In the middle, we put the dorsal root ganglia. On the other side, we put these microtubules or loaded straws with nerve growth factor. And we show that we can implement the chemotactic design in vitro by seeing that all of these things grow if you capture them at the right time. I'm sorry that you can't see the blue very well, but essentially 100% grow this way and not this way because of the chemoattractive thing. This was just to demonstrate that you can implement a chemotactic strategy. So imagine then if this is the end of the nerve, you can attract it to go this way so that you want regeneration to occur in that direction. So this is just in vitro principle showing that you can implement that strategy. So with that in mind, we made different gels containing laminin alone or nerve growth factor with these embedded systems, or containing both with an autograft, with just saline. And with all those controls, we did a 10 millimeter nerve gap in a sciatic nerve of a rat. Now with the 10 millimeter gap, it's a polymer guidance channel. So this white thing you see here is a polysulfone semi-permeable guidance channel that you can commercially buy. So if you just have a saline-filled channel, in 1 in 10 or so, or none, will you actually get some kind of cable forming. So it's a model where to get regeneration significantly, you do need to do something with it. Now this tells you in how many of these animals, if you had 100 animals, what percentage of those animals, this is percent on the y-axis here. So if you had an autograft, in 100% of the animals, you get a cable forming across a 10 millimeter gap. Now that's the clinical standard. This animal model, this is just to calibrate you to the animal model. Okay, so 100% with the clinical gold standard, you get a cable forming. Now if you just did PBS, in the channels, you get about 20% of the cases. If you just put agarose or just put empty tubes with no NGF, we put laminin, just NGF, containing both NGF and laminin, you get 100%. So if you do just laminin or just NGF, you get some gains over saline, but not dramatic. But if you put both, you match the performance of autografts by this criteria, a very crude criteria. Is there a cable or is there not a cable? Okay, and so that's the beginning. So the question then is, what is the nature of this cable? Is it a useful cable or is it just stuff? So if you do histology and cut transections of the nerves and stain with osmium tetroxide, which stains for myelin, you can start to look at the structure of what you get. And this gives you, this is your native nerve, this is your autograft, this is when you have laminin and NGF. And what you're seeing here, again, for those of you who are not used to looking at nerves or cross-sections, you see typically this black thing here is your Schwann cell that is always around one of the circular thingies, and you're staining essentially for myelin in the middle of the bridge. Okay, so this is this is at 5 millimeters in the middle. This is how things look. And you can count, you can do various kinds of analysis. You can count the numbers of myelinated nerves. Now this does not allow you to count the unmyelinated nerves. To count unmyelinated nerves, you need to do transmission electron microscopy. It's really, really painstaking to do quantitative transmission electron microscopy. But, so you can do various kinds of analysis looking at this, and we did. So we looked at two things that I'll talk to you about today. One is the total number of myelinated axons. On average, we did, I think, 12 animals of each or something like this. And this is published, I can give you references. And then the density of myelinated axons. So if you just look at the number of myelinated axons, if you just use laminin or NGF by itself, you do get a significantly different effect than using saline or just the gels plus empty drug release vehicles. However, if you include both laminin and NGF, or if you use autographs, in this case you get, they're similar, there's no difference between these two, but they're not as good as your native nerve. Your native nerve has about, in rats, in sciatic nerves, you have about 8,000, 9,000 myelinated axons. You're only getting about 6,000 or slightly less with both the autograft and your engineered construct that has both laminin and nerve growth factor. So there is room for improvement. If you look at the nerves that grow back here and say, well, you know, are they normal looking or what? And you do a density measure, which is number of myelinated axons for a given area. You actually, they're all the same. So the nerves that you're growing here are similar to these nerves, except they're just smaller cable. And that's what this data is showing. Again, you need both laminin and NGF in your engineered construct by this measure, and you match autograft performance. So the moral of this story is you can match autograft performance, but you're still not as good as the normal nerve. So you can do some kinds of behavior analysis, and I won't show you all of this data. One of the ways you can do this is the so-called sciatic functional index. And we basically mount a video camera under a glass table, and we watch the rat walk. And you can measure the toe spread to get a sense for functional recovery. Okay? So we did this 2 months after our implants. And you can measure this, and it's kind of painstaking, and graduate students don't like it, but the good thing is, you know, we get popcorn, and they have a nice screen, and they watch with it. But so you can do some kinds of analysis, and essentially when it's injured, the toes curve up, and as it gets better, the toe spread increases. And the way the functional index works is the lower the number you get, the better the nerve is. So if you look at the data, actually, at 2 months, with the autograft and the laminin and NGF containing fiber, you have a significant improvement, but not really impressive. We need to wait 4 months or so, I think, to get really functional recovery with this 10 millimeter gap, and that data we don't have yet. But you'll see that although we got all these numbers of myelinated axons and things, really the functional recovery is not all that impressive. It is significantly different. And all the things I said earlier are true in the sense that the autograft and the laminin and NGF channels are good and they match. But you know, it's good to write a paper, but I'm not jumping up and down about getting into the clinic with this kind of results. Perhaps if we waited 4 months, there would be more dramatic, but still not as good as we'd like to go into the clinic and replace autografts. Okay, so we need to do something else. But the moral of the story is that we are able to match the performance of autographts in this model. So that is true, but there's work to be done. So that's what I say here. Just to summarize this part, you can present large extracellular matrix proteins like laminin in 3D using a tissue engineered gel. And I didn't show you this data. We have all sorts of studies to show that the improvement in growth you see in these gels is actually receptor mediated. It's not because of changing the porosity or the mechanical properties of the gel. You can set up systems to have trophic factor gradients that can elicit directional neurite extension. And these strategies work across a 10 millimeter gap. But if you have a larger gap, when I saw this data, I did not have the confidence that we would get robust regeneration across larger gaps using such systems. And so we started exploring 2 alternate systems. One of which was to use to introduce into the system what I'm beginning to call for the last 2 years anisotropic elements. The idea is to provide directional cues and use that to get regeneration across longer gaps with the idea of it's like giving a biscuit to a dog and moving the biscuit along. So the idea is, can I put in cues within this bridge such that there is some directional element that makes the nerve want to go further than it would if it did not have the directional element. So one of the ways we're doing that is by making three-dimensional hydrogels that have gradients of embedded proteins in them. And this is just a cartoon showing you that you can make a gel which has increasing amounts of protein across the gap. And there's a paper that's just been accepted, and I don't have time to show you all of that data, but the data is really interesting. If you take laminin and you generate various slopes of this data, just to summarize this paper that's in press right now, it turns out that when you have a gradient, even though the absolute amount of laminin you have in a uniform gel, if you compare the laminin in a uniform gel where laminin is distributed uniformly versus a gel which has a gradient, even though the highest amount of laminin you have in the gradient is lower than the laminin you have in the uniform gel, you get faster nerve growth and better growth in the gradient systems than compared to uniform systems. So it turns out that the growth cones seem to respond to change of concentration of laminin much better than the uniform concentrations of laminins. And we took care to see that we were below the saturation of the laminin concentration of the uniform. So we did that experiment, and so we're within the saturation level. So I'm very encouraged by that, and we have actually in vivo data that is, next year I can present to you, or next time we see. But this strategy I think will work well. The other strategy is to implement the contact guidance. Remember I told you there were three things: haptotactic gradients, chemotactic gradients, and the last one is contact guidance. This we're very excited about, and I hope you'll agree with me as to why when you see the data. So when you have a longer gap, I told you that you have this fibrin cable form that lets Schwann cells migrate in. It turns out that when Schwann cells migrate in and they have these fibrin cable, the fibrin goes away, but the Schwann cells deposit laminin. And you have a structure called the bands of Bungner, B-U-N-G-N-E-R. Bungner, there's probably two dots there that I don't know how to do on my computer. But the bands of Bungner form, and they help the nerves, and the nerves follow those bands of Bungner when they regenerate. The question is, when I have a long nerve gap, can I recreate the bands of Bungner by some other artificial strategy? And if I do, will I get regeneration across a long nerve gap? Okay, that's the question we asked, and we said about designing a system to implement that. So the way we did that is we used electrospinning, and several speakers introduced that, and Steve Badlack introduced this. So normally when you electrospin a polymer, or you can electrospin anything really if you work hard at it, you usually get a mat because you can't control the polymer that comes out. I don't know how many of you are familiar with electrospinning. Essentially you have a fine nozzle and you have a high voltage electric field, and you pick your solvent and your polymer such that the polymer is propelled by the voltage to strike a target that's conductive. We built a fast rotating drum on the other side, and so we have aligned fibers. It's an old trick in polymers, people know this. But we just put it to electrospinning to get aligned fibers, and we make mats or films of these electrospun fibers that are aligned. And the thickness of the film we can control by just the time that we spin. And right now we spin for a time that gives us 10 micron approximately thick films that we can actually handle. So if you take such oriented electrospin fibers, and the first data I'll show you is with a non-degradable polymer that we started playing with, PAN-MA, which is polyacrylonitrile methacrylate. So we made these fibers that are about between 400 to 600 nanometers in diameter. Okay, 400 to 600 nanometers. We have now, our goal actually in the future experiments, I'll talk to you in the end about this, is to make sub-100 nanometer fibers. We have a couple of interesting techniques to do that. But essentially, if you take a film of these electrospun fibers that are oriented that way, I should put that picture here actually, I put it in the ground, I forgot to put it here. If you plate neurons on the surface of these films, you get this beautiful alignment of fibers, and you can, it doesn't take a rocket scientist to know that this is But if you look more closely, what happens is the colors here mean something. The red is neurofilament 160, which stands for neuronal processes, and the green stands for Schwann cells. And this is here the green stands for laminin. Essentially, we see Schwann cells migrate out first from the DRGs, the dorsal root ganglia, and then nerves follow. We never see a nerve. Very rarely see nerves out by themselves without a Schwann cell. We also see a lot of laminin deposition. Again, sort of recreating the bands of bone nerve. And these are, we've done both adults and P1, postnatal day 1 DRGs, because you can say, well, embryonic DRGs grow well, and you want to repair an adult nerve, is it the same, So we tested the adult DRGs, and they grow as well. So these films are very nice. The question is, how do you make a 3D matrix out of this? And we do that by layering these films. Okay, so the idea is you have a guidance channel, and you can layer, I think my point was dying on me. I have another one I'll give you. So that you can layer these films and put a hydrogel that I talked to you about earlier with agarose in between to control the spacing of these films. And we did that with the orientation in the longitudinal axis, right? So if you do that, you can make, this is the actual photograph of such a scaffold. So you can see the gaps in the film. And in between, we put the gel that I'm showing you over here. So yeah, the bottom two panels show you the principle that I was talking to you about. Now, although there's a very interesting, oh, thank you. Hopefully I won't break this one. So the principle is this. So it's been, Paul Letourneau demonstrated this really beautifully, he's in Minneapolis, that growth cones choose, if you give them a choice of two substrates, they choose the substrate they prefer. So if you have three substrates, A, B, and C, and let's say they prefer C most, B next, and A next. If I just give them A and B, they choose B. If I give them B and C and they like C better, they choose C. Okay? So they always take the preferred route, and that kind of is logical. There is a similar property when you think about 3D, and this is, I encourage you to think about anisotropy in the Z direction. I call this the Z directional anisotropy. If you take a fiber and embed it within a gel such that the neuronal process in this case prefer to grow on fibers, they will latch onto your fiber and grow, but when the fiber ends, they don't mind just going on into the gel. Okay? So I'm convinced that, judging from the results earlier where we made agarose gels of different kinds, that this is the way to go, that we need to embed things within. We also know when you culture 3D cells in 3D, many times when cells sink to the bottom, they grow beautifully at the interface of the gel and the tissue culture plane. And we're kind of implementing that with these fibers. So this is just to show you that you can get things latch onto the, this by the way is 25 microns, so this is a single nylon monofilament suture, cano suture that we were doing surgery with. So this is clearly not a nanometer fiber, but just to illustrate the principle. So we took these things and we created a gap of 17 millimeters across. So the 17 millimeters is a really challenging gap in a rat sciatic nerve. You pretty much have no more room when you do surgeries. And you get 0% regeneration. I'll show you that data. So we took a longer tube, you can see this is a little bit longer, and put the stacks of oriented film within, and then, bridge the nerves. And we used autograft as a control, and we also did the 3D construct, and we also just did a construct by itself with nothing in the middle, right? So those are our three things. No laminin, no growth factors, no proteins, just the scaffold, all right? So the question was because we wanted to just test the contact guidance, not the other things. So here's the data. This is a very complicated slide, but I'll take you through the relevant things. This is just from a pending publication. So if you have an empty channel, nothing grows. Okay, so this is proximal, this is distal, it's completely empty. If you have the red here is nerves, and the green is Schwann cells, and if you see yellow, it is both nerve and Schwann cells. Okay, so you get nerves growing across, and this is, remember, one section, so this is not a 3D thing. So this is just one optical section across a 17 millimeter gap. It's actually a montage. And then you see a lot of Schwann cells coming in from both sides. And you see lots of yellow if you look at both proximal and in the middle. Or here you see lots of yellow. If you looked here by itself, there's no yellow, just Schwann cells, or only green stuff, no red, so there's no yellow on this panel here. So you get beautiful nerves. I shouldn't call it beautiful because I'm quantified nerves, but it is beautiful in a sense. You do get regeneration when you across. Now remember, this is without any exogenous proteins, no trophic factors, just using the scaffold. If you look more closely, you do recreate in the autographs this so-called bands of Bungner, where you have Schwann cells and laminin. So this is a counter stain for Schwann cells and laminin. So if you look at yellow, you see Schwann cells. We do a stain for blood vessels. There are lots of blood vessels inside. And there's an emerging story that Steve Stice tells us, and we're very interested in, that the whole angiogenesis and neurite extension thing, there's a lot of interesting overlap. And Steve mentioned it a little bit last time. But we do see beautiful blood vessels as well. We see GAP43, which stands for regenerating fibers. So we have very extensive studies showing histologically that we get very nice regeneration. If you look distally in the muscle at the end, you can stain for things like alpha-bungarotoxin, which is a cholinergic acetylcholine receptor stain, to look at neuromuscular junctions. So again, the things are color-coded here, the labels. So the red is alpha-bungarotoxin, and you can see red in the muscle. So functional neuromuscular junctions have formed after the regenerating nerve has gone all the way across. All right? So again, using the nanoscaffold. And there's co-localization of alpha-bungarotoxin and Schwann cells, which is also true in neuromuscular junctions. We've also done electrophysiology to look at the nerves to see if they're functional. So before we sacrificed the animal and did all of the histology, we did electrophysiology. And the way we did that was we stimulated the nerve that regenerated distal to the implant. So if this is the proximal, this is the scaffold, and this is distal, we stimulated here and recorded in the dorsal or ventral root. Okay? The idea being that whatever I hear in the dorsal root is sensory fibers that have regenerated. Whatever I hear in the ventral root will be the motor fibers that are regenerated. And you can do all sorts of electrophysiology, including conduction velocity, the latencies, the threshold of stimulation, to get a sense for the nerves that have regenerated. So this is the mixed nerve. This is sensory. This is all of the nerve together. This is just recording with the dorsal root. This is just recording with the ventral root with motor neurons. And the long and short of the story is that the peaks are much more broader, so you have a much more slower, and the range of diameters is probably smaller in the autograft and in the regenerated case. We've done both. This is normal autograft and nanoscaffolds going this way. So in 3D constructs, or what we call nanoscaffolds within the nanofibers, you get broader peaks. In the nanoscaffolds, you seem to actually get a little bit better both sensory and motor, whereas with the autograft, you don't get that many sensory nerves going back, just motor, it seems. So I don't want to explore that too much, but essentially the nerves that we grow back are functional, and definitely autografts and nanofibers match. Sorry about the quality of this, but we also have behavior testing called grid walking, and we measured latency. Again, the long and short of this is that both autographs and nanoscaffolds are similar, but there's a long way to go. In grid walking, you actually measure slips. The number of times your foot slips, so the lower is better. In normal, you don't slip as much. In autograft and nanoscaffolds, you slip less. If you just have saline construct, you slip a lot when you're trying to walk on a grid. I'll show you some movies with the CNS about that test. But both electrophysiologically and behaviorally, the nanoscaffolds and autographs work together. So summarizing just this part, contact-guided nanoscaffolds alone, without any exogenous proteins, are capable of matching the performance of autographs. Now this has, I have not seen any reports of such a finding across a gap as challenging as the one we've used. And the next step really is, can we exceed the performance of autographs? Because I told you, while autografts are the clinical gold standard, I think we can exceed the performance of autographs if we try. So we're putting together a 50 millimeter rabbit model in the lab with the help of our veterinarian at Judge Tech. And we are proposing to coat the nanofibers. First of all, we're going to make sub-100 nanometer nanofiber scaffolds. So the idea is to get on to the structural scale of extracellular matrix fibrils. So when we think of extracellular matrix, generally we tend to, in tissue engineering, think of them in their biochemical nature, in the sense that we think of them as proteins that interact with receptors and work. But there is a structural element, and Steve Bartlett's work really shows us very clearly that there is a structural element to them. And what we'd like to do is mimic the structural element of some of the extracellular matrix molecules using the nanofibers that we're making. We have a couple of different ways to make commercially viable sub-100 nanometer fibers that are not electrospun. Electrospinning is actually very hard to do on large scales. And we have actually other technology. We published some papers using layer-by-layer coating to put 10 nanometer coatings on these fibers of laminin. And we can also embed within the scaffolds the nerve growth factor releasing or BDNF releasing fibers. And the goal is we really want to see if we can beat the performance of autographs. Now that we have a handle on how they work, beat the performance of autographs across a 50 millimeter nerve gap. If we do this, I think we can go to the clinic. And I think we'll know in 2 years whether we can do this or not. So that's where we are. So that's my conclusion. I think if tissue engineering strategies offer a great promise to bridge long nerve gaps in the PNS, I think the way we think of tissue engineering is it's a way of thinking and implementing strategy that we derive from our understanding of biology. Just want to thank Jun, who did all, he's actually a professor now at graduate student of mine, Professor Stevens, talented postdoc, Yamke Kim, collaborator Satish Kumar from Polymers. This is my group, this is our building, this is biomedical engineering, and IBB, where Bob Needham is over here with the bridge. My lab is over there. So I'm funding from GTech and NIH. So I'd be happy to take any questions you have.
Thanks, Ravi. Thanks. This is open to questions. Can we have the lights up? Who controls the lights? Somebody must. Okay, any questions out there? Yes.
Time lapse between what? The trauma. Yeah. Yeah, so you ask a good question. So in all our cases, we resect the nerve and immediately do the repair. So we have not looked to see what happens if we wait. We have not done that. I know that if you crush a nerve upstream or wherever, and then you resect, you actually get a better response because you condition the Schwann cells to respond in a pro-growth way, and then if you do it, you get actually a better response with nerve crush. So sometimes people do a crush, and then they do the transection, and then they do repair, they get a better response. But I don't know the answer to what happens if you create a nerve gap, wait for several days, and then do it. I do know that you do not get too much retraction in the spinal cord or here, which is encouraging. Sorry? Oh, cell phone. Somebody's cell phone is ringing. Somebody's cell phone is ringing. Yeah. So I do know that the proximal nerve endings do not retract very much, which is good for us. So that we may have some time to go do this. Yes, buddy?
So the difference, the fact that in the central nervous, the fact that in the central nervous system scarring occurs, but in the peripheral you get no scarring. It's really very interesting. Does anybody know why there's such a huge difference?
Yeah. So we know that the cellular constituents of scar and the cells that make scar are very different because the cells in the vicinity of the injury are different. The Schwann cells, actually there is in autographs, there is a group in Florida that's the basis of this company, Axogen. There are chondroitin sulfate proteoglycans in the peripheral nerve autograft. That's another reason not to use it, and that's why. If you digest those grafts with an enzyme to digest them with the chondroitin sulfate proteoglycans, which are the inhibitory parts of CNS scar, the autografts work a little bit better than if you did not. So there are inhibitory things, they're just not as much as there are in the CNS. But the principal reason, I think, is that the Schwann cells put out so much trophic factor and so much NGA, and there are no oligos and others that make a bunch of the negative stuff. If you ask me evolutionarily, why is it that way? I think it's speculative. Why do Klingons have the spinal cords regenerate and not us? And I think my understanding is that we've placed a premium on correct connections in the CNS, and it doesn't kill you. A spinal cord injury rarely kills you. So the idea is you wall off the injury, you take your losses, you do not. So we don't know if we were to successfully regenerate spinal cords, if that would be a good thing or not. We just don't know because we've never done it. So, but I'd like us to know. so yeah.
Down here, can we get a microphone to?
Thank you for the excellent presentation. And I have a question. Concerning the nerve autograph. Does this apply widely on patients?
Yes.
Is it widely applied?
It is, yes.
So
that is the only thing you can do.
So far, but
There is, in the market, the channels are available. So there are companies that sell just the polymer tubes.
so it's available like on shelf on the market?
Yeah.
Autograph.
Not autograph.
What?
Autograph is auto for the
That's
same
what I mean,
patient.
yes. So
The other thing that clinically people do are use the guidance channels, just the polymer channels. So those are people are selling.
no, I'm talking about the nerve autograph.
I understand.
I'm talking about.
Yeah.
So he compromises the site, like the location of the same patient, that's right?
That's right.
How far is the compromise for the location that the graft from?
So usually you'd use the sural nerve, and so there are complications with that. That's what I was trying to say earlier. But so depending on the nerve you're trying to repair and how important it is, people are willing to lose the
So
sural
we are
nerve.
ready to lose some sensation, of course not motor, but sensory.
That's right.
But what about the pain experienced by the patient later concerning this is following the nerve injury or the passing pain?
That is right. So you have neuroma possibilities. It is painful. That's what I say in my grants. That's why we do this.
Then still they ask for the autograph.
Yes, because in cases like brachial plexus repair, in vocal cord,
This should be priority.
that's right, exactly.
Thank you.
It's not great, but
The best possible solution.
that's right.
Other questions?
Yes, sir.
In the back there.
I can repeat your question.
No, those don't work.
What is the type of polymer used in the nanoscaffolding?
So we use polyacrylonitrile methacrylate copolymer, PAN-MA. It is mostly polyacrylonitrile. We've also made, I didn't show you all of that data, both PLGA and PLLA nanofibers. PLGA nanofibers are no good because they shrink by almost 50%, but the PLLA fibers don't. So our next generation, in fact, I have a grant that we just submitted where we propose to use a degradable thing, so we use PLLA fibers. And we also have another technique to make these nanofibers not electrospinning.
Okay. If no other questions, then I think we're going to take a 10-minute break.
An announcement.
Yes, Dr. Gadd.
Please, the poster presenters are kindly requested to go and remove the posters because we need room to prepare for the roundtable discussion.
The roundtable's in here.
So you can have sugars that are not sulfated at all, and they would be chondroitin. You could have 4-sulfated, which is on this sugar. There are 2 sugars, the glucuronic acid and galactosamine. So this is a disaccharide that repeats. So you can have a 4-sulfated sugar here. You can have a 6-sulfated sugar. You can have a 2,6-sulfated sugar, where 2 is over here and 6 is over here. Or you can have a 4,6-sulfated sugar. So these are the, some of they have other names. Instead of calling them by position, you can call, this is chondroitin sulfate A, this is C, this is D, this is E, this is B. This is dermatan sulfate, by the way. So this is chondroitin sulfate B, which is 4-sulfated sugar. So the nomenclature also is a little bit messy, but this is the diversity of sugars that are known already from chondroitin sulfates, from cartilage and other places. So we asked this question. So are the gags upregulated in injured brain, and if so, which ones? Just the sugars, okay? And they never exist by themselves. They're always with the protein core, and the protein core may also contribute to inhibition. But in this talk, I'll just focus on the sugars. And if they're upregulated, are the upregulated sugars any more inhibitory than the other ones that are normally present? And I'll show you a couple of different ways of using quote-unquote tissue engineering strategies to answer this question. So we used a technique with the help of Vince Haskell at the Cleveland Clinic Foundation, who studies cartilage, by the way. And it's a technique to analyze very small quantities of sugars in a quantitative manner. And the technique is called fluorophore-assisted carbohydrate electrophoresis, or FACE. Okay, I didn't put that in here. But it's FACE, and I can give you references for this technique. Essentially, you take your gags, you digest them away, and you can run a gel on these. It's tricky, but it can be done. We do this in our lab. And you can have all sorts of standards, and you can run, and we looked at the sugar profile that's present in a normal adult cortex and compared it with scar tissue alone from the brain. So we generated scar tissue by making an implant of a film of nitrocellulose and explanting the film with scar tissue alone. Okay, so we had no normal tissue. And we looked at the sugar profile of adult cortex and just scar tissue. If you did that, here's what you see. You see in normal cortex, 91% of the sugars, the chondroitin sulfates, are 4-sulfated sugars. You have very little 0-sulfated, you have some 6-sulfated, okay? In the injured cortex, however, and this is scar, okay, this is not injured cortex really, this should be scar tissue. In scar tissue, you suddenly see 2-sulfated, you see 4, but it's a lot less as a percentage. You see a lot of 6, and you see for the first time a 4,6-sulfated sugar. Okay, this is a dual-sulfated sugar. Now this is in scar tissue. This is percentages, so and the total amount of GAG also is higher here than here. So both the amount increases and the profile of the chondroitin sulfate GAGs in the CNS scar tissue is different from the normal adult brain. So the question then is, well, are the things that are showing up in excess amounts like the 6 or the 4,6 or any of these, are they any more inhibitory than the others? To test that, we used our tissue engineering sort of strategy. Normally in the PNS, we were using these kinds of gels that I showed you earlier in the middle of the bridge to get growth, to permit growth. So instead of doing that, what we did was we coupled the gags to the backbone of these gels. And the reason we had to do this in gels and not in 2D is because if you take these sugars and immobilize them on a surface or absorb them on a surface, they're very, very hydrophilic. So if you just absorb them, they lift off and go into the solution. If you immobilize them, because they're so hydrophilic, cells don't attach to them. So you can never really, if you don't have a neuron attaching, you can never really ask the question, is it inhibitory to a growth hormone, because it never grows, because it never attaches, right? So to do that, we use this 3-dimensional system that we had developed for peripheral nervous system, and we coupled gags to the backbone of these gels. And we compared what happens when you have equimolar amount of different gags in such a gel, and you put neurons inside, so that you get a sense for the potency of the gel. We did not put in the gel, which we could have done, the ratio of the gags we saw. We put equimolar amounts. And if you put equimolar amounts, we asked the question, because there's no question of attachment now because the neurons are trapped inside the gel. So you're able to separate the issue of growth cone inhibition and initiation of growth or attachment. Oops. So if you do that, again, this is from a paper that came out last year in Molecular Cellular Neuroscience. What we show is that the chondroitin sulfate E, which is the 4,6 sulfated GAG, is potently inhibitory, as inhibitory as the entire CSPG, the chondroitin sulfate proteoglycan with the protein core and the sugar. So that's what this bar is. This is the average neurite length, and we do some software analysis to figure this out. But trust me on this. But so it's believable and repeatable, and it's double-blinded stuff, and we used a couple of different measures. So the moral of the story is that both CSE and agacan are potently inhibitory to neurite extension. B, which is 4,6, is also inhibitory but not as inhibitory. We do not see any inhibition from the 6-sulfate. So CS4, which is present a lot in the normal brain, but 4,6 is potently inhibitory, as inhibitory as the entire molecule. Okay. And nobody really had shown this. Ours was the first lab to demonstrate that even 4,6 existed in scar tissue. Nobody had even looked for it. So we were happy to report this in the last year. So if 4 and 4,6 are inhibitory, do they inhibit growth or initiation of growth cones? Because we cultured these in gels, the reviewers might ask the question we taught, and so we did this experiment, that if you put this in the cell, how do you know if they're just stopping the beginning of processes, or if processes begin and then they don't grow when they see this thing, right? So we needed to separate that issue. And to do that, we created 3D interfaces to sort of mimic the interface that I showed you earlier with scar tissue. And to create an interface, we did a little trick. We put in the middle of 3 layers of a hydrogel, we put in the middle the DRGs, or dorsal ganglia, and on either side, we put a gel that had the different glycosaminoglycans on the other side. Okay? And then we asked the question, once they started growing, what happens to the growth cone when they hit the interface? Now this more closely mimics the interface of the scar tissue that regenerating growth cones hit. And so that's the question, and then you can lay these things on the side, and you can observe from the top and see what's happening. And if you see this, normally if you have on both sides the same gel that is permissive, no gags, they actually cross pretty well. This you can see optically at the interface. If you just layer 2 gels, to rule out the fact that just does layering cause inhibition, we did this control that if you just layer gels, they do cross. I mean, you get about 80% of the processes crossing. Okay? But 20% gets stopped due to surface tension effects at the interface. But 80% do cross. If on the other hand, instead of having a gel just like here, if I put a gel that has the chondroitin sulfate GAG on this side, this is what happens. Boom, nothing crosses. Right? So this is more like the scar tissue where you get things to grow, they hit the interface, don't go to the other side. Again, clearly demonstrating this is not an inhibition of initiation of growth cones, but really inhibition of growth cones after they grow. So this is one strategy of using sort of tissue engineering to make a better model of scar in vitro, in a sense. The other thing we did was, we wanted to understand, because nothing is known, we don't know what the receptors are, we don't even know if there's receptors, we don't know why they're inhibitory, we have no idea. And so we have some efforts, I have a postdoc fishing for receptors, but that's a risky business. But we did some dynamics studies where we made some movies, and I did not include this movie, but I'll show you other movies, of growth cones growing in gels that had both, in this case, CS4 and laminin. And we analyzed how the growth cones were actually being inhibited to get a better sense, perhaps, of the mechanism that might be involved. And it turns out that when growth cones are growing, they follow a cycle. They grow. First they explore. They send out these lamellipodia or filopodia where they're exploring different things. Then they're actually displaced, and then they rest for a while. And this cycle of searching, growing, stopping repeats when growth cones grow. They're just not always growing. So they search, then they actually displace, then they stop for a while, then they search some more, they displace, and then they stop for a while. So this is a cycle of growth. And so they go from stationary to searching to growth, stationary, searching, growth, and the cycle repeats. So we looked at movies and analyzed how this works, and this work is in review right now. We show that the time required for one cycle, when you look at these movies, the time required for one cycle of neurite extension is much higher when you look at chondroitin sulfate or C4-containing gels compared to laminin, which they love to grow in, compared to a gel which has nothing, neither laminin nor CS. Okay? So it turns out that something between the cycle thing, at some phase of the cycle, they're taking a longer time. The periodicity of growth, if you will, is much longer, or the frequency is lower with the inhibitory gels. If you look at the growth during the growth phase, when they're actually moving, if I look at the rate of displacement of the growth cones, it turns out actually that these two are, this is a little bit lower, but not really that different. But this significance has to do with laminin. That in the laminin gels, when they're actually growing, they grow much faster. So there's motoring. They're happy they're growing much faster because they're actually moving. And with the CS gels, we don't see a big difference. We see some, the mean is a little bit different, but we don't see a big difference in the actual growth. Okay? But if you take the average growth, which includes the stationary periods, you get a laminin because it's both including the actual difference in growth rate and the displacement as well as the resting periods. Again, you see with laminin they do much better. In CS, it's pretty, it's lower but not pretty close to the agarose, which is interesting. So we did these experiments because we wanted to understand the mechanism behind CS gag inhibition to give us a clue as to what to look for in terms of signaling. And we have a whole host of, we have a postdoc and a couple of graduate students looking at calcium signaling and Rho GTpases and actin filaments and all of this, trying to get a sense for what it is that the gags may be signaling at the end of the growth cones. But what we do know, just to summarize this part, is that CS gags, the profile is differentially regulated or upregulated in the injured cortex compared to the adult cortex. The upregulated gags, specifically 4-6, is potently inhibitory, and they affect growth cone dynamics and increase their resting period generally. That is why they're shorter when the gags are present. The resting period is much bigger, and the displacement is actually not affected. So it is not perhaps mechanically somehow different. We've done that analysis also. But the gag gels are not any stiffer than the other gels. So I'd like to move to the next phase of my talk, which is to see, well, we know all this, but how can we use this information to increase regeneration? So as I told you, the blunt instrument right now is to use this enzyme chondroitinase ABC. And there's a company called Accorda Therapeutics in New Jersey that is trying to make these enzymes and use them. And I'm very skeptical about this therapy, and I've told them so on a phone conference, didn't make them happy. But the reason is that chondroitin sulfate, CSPGs play a very important role in the adult brain. They exist for a reason. They make structures called perineural nets. Okay, and this is just being understood now. There are these nets around neurons called perineural nets. And it turns out that the plasticity in the brain, which we're just beginning to understand better, is perhaps regulated by the presence of these perineural CSPG nets. And in the hippocampus, for example, where you have more plasticity because of memory and such, you have modulation of CSPGs. And in other areas where you don't want to get plasticity of growth, you have more CSPG. So in my mind, applying chondroitinase ABC is a very blunt instrument because it indiscriminately digests all CSPGs. And I think it will have some deleterious consequences if we were to do this clinically. So our goal is that if you understand more specifically why CSPG in scar tissue is inhibitory, and understand perhaps if a certain constituent of scar is responsible for the majority of the inhibition, I would much rather have a more finer knife where I inhibit just that component and not all of CSPG and digest it away. So that is the approach. So one way to do that is to use small interfering RNA. Again, both Pat gave a very nice introduction to that. And then I'll show you a couple of different approaches where we could perhaps make, so you can, if you think scar is the big problem, the approach is you can make scar go away, that is what chondroitinase ABC does. you can only delete the inhibitory bits of scar. That is one approach, and I'll show you some data. Or you can make the growth cones less sensitive to scar. If you understand what it is that these growth cones are feeling when they're seeing scar, and you can somehow modulate that and make them less sensitive to scar, perhaps you can make them go through. That is one approach. Or perhaps you can downregulate the entire system using MP therapy, and I'll show you some data on that as well. So these are the different approaches theoretically you can adopt. So I showed you all this gag data, and the question I ask you is, what are the implications? So one way to downregulate GAG specifically is to figure out what are the enzymes that make GAGs. And is there a way to downregulate the enzyme that makes 4,6-GAG, which I showed you was very inhibited. To do that, I'll tell you a little bit about chondroitin sulfotransferases, or chondroitin sulfate sulfotransferases. And again, all of you are interested in cartilage, if this applies to you also. So it turns out we know the family of enzymes that make CSSTs. And so we targeted the chondroitin 4-sulfate 6-O-sulfotransferase, or C46ST as we call it, because this is responsible to put the 6-sulfate on a substrate that already has 4. And that's what its method of action is. So we asked the question, can we, or would it be viable for us, to try to downregulate this enzyme in some specific way, downregulate 4-6 formation, and will that then be sufficient to get better growth across scar? So that's the approach. And today, unfortunately, I have just data to show you that we can downregulate this enzyme, but I don't have the functional data because those studies are still ongoing. So to do that, what we've done is we've designed small interfering RNA constructs against the 4,6-sulfate enzyme that is responsible to put the 6 sugar on the 4 to make it 4,6. And I'll show you in vitro. So we made 4 different constructs. Again, just to tell you that this is how siRNA works. It works by degrading the mRNA for the target protein. And it's more efficient when it works. It's more efficient than ODNs because the thing then can go and degrade another one. So you don't have to get the dose matching exactly right. Because in ODN or oligodeoxy technology or antisense technology, it binds the mRNA, but then you have to match the dosing to get it all. So this works much better, I think, because it degrades it than it binds. So what we did was we made 4 different constructs. We have an in vitro system with astrocyte culture where they make 4,6 and 4-sulfated GAG. We showed that earlier with face analysis. And so we asked the question, can we downregulate the 4,6 enzyme in astrocytes in vitro as a test bed before we try it in vivo? And this is with 4 different constructs. If you focus on this part here, so the number here is construct 1. They were either given siRNA once or twice. And so this constructs 3 and 4 seem to be most effective. And we stimulate the astrocytes in vitro with TGF-alpha to make them make these CSPs. So in an in vitro assay, we have identified siRNA constructs that are capable of downregulating the 4,6 enzyme. And now we will ask the question, well, will the scar tissue, after you knock out this sugar alone, will it be, less inhibited? But the other curious thing we noticed when we did this is, and nature always, plays a trick on us. When we downregulate the 4,6 with these constructs, we see an upregulation of the 4 sugar, which is also inhibitory. So it's like the cell is saying, okay, if you're going to knock this down, this is what I'll do. I'll increase this and let me see what you'll do. So, and this is, the people who do knockout mouse and things know this. Every time you downregulate one pathway, something else goes up and tries to compensate. And that's what we're observing here. It's good we observed it early with our extensive in vivo studies. So what we're proposing to do now, when we observe this, is that we will actually do a combinatorial knockdown of both 4,6 and 4 when we go in vivo or in vitro look at the consequences. And that's where we are now. We've made constructs against 4 also, and we're testing them now in vitro, and that's what we'll do when we go in vivo. So just to show you, we've demonstrated that we can knock down the enzyme, and knockdown leads to upregulation of another inhibitory mRNA. It's not as inhibitory as this one, but also inhibitory. So we would like to knock both of these down, I think, when we go in vivo. But this could be an approach to more selectively get rid of only than inhibitory and not digest away the whole CSPG. We think it's a more elegant way to go. The other strategy I'll show you is using modulation of small G proteins, or rho GTPases as they're called. So what are rho GTPases? And they've been studied extensively in fibroblast migration and migration of a whole bunch of cells. In the context of the nervous system, they're responsible for filopodia and lamellopodia. So when you see pictures of the growth hormone in a textbook, it looks something like this, where you have a flat part, which is your lamella, or lamella being a layered structure, with filopodia being the individual ones that go like this. And so the filopodia, the searching, it turns out, is regulated by this molecule called CDC42, and the lamellopodia by RAC1, a protein. And so they're basically modulators of the cytoskeleton. And you can imagine that if I'm growing, I want the cytoskeleton is responsible for the architecture of your growth cone. And if you're moving, you need this to work. So that's what these two small G proteins do. And the way they work is, in the GTP state, they're active, and the GDP state, they're inactive. And we have access to mutants of these proteins that are permanently in the GTP state. So we have, and they're called constitutively active proteins. So if I put these proteins in, no matter what the state of the endogenous proteins, they're always on the on state. So if I put constitutively active CDC42, it'll always be active. It cannot be switched to the GDP state where it's inactive. All right. So we have access to the mutants of all the three, rho A, CDC42, and RAC1. And when we do this, we did an assay where we made lanes in 2D of CSPG. Now, this is not the gas. This is the whole CSPG. So that doesn't lift off because it's a big protein. So if you put macrocellulose at the bottom, we can put CSPG in the middle, and nothing grows there, and this is laminin. So the thing at the bottom tells you where CSPG is and where laminin is. The black parts is where CSPG is, and the light parts is where laminin is. So if I do that, I get occasionally some growing across, but for the majority, you don't get crossing because they don't like CSPG. We've already established that. This is just another experiment establishing. But if you hit them with mutants of various things, and I won't go through all of these, but with two things, with CDC42 and RAC1, you get a lot more crossing of these things, and you can count the crossings, and that's what this data is. This is also published, I can give you a reference if you like, showing that constitutively active CDC42 and constitutively active RAC1 both increase or make the growth cones less sensitive to CSPG. Notice that in this case, we didn't do anything to the CSPG. It's still the same CSPG. We didn't digest away the sugars. We didn't downregulate any of the sulfate sugars. So clearly, the growth cones, we've done something to the mechanics of growth or the sensitivity of these growth cones to the inhibitor by mucking around with the small G protein. So this could be an approach, and I have a student pursuing this in vivo right now. But what we do have sort of dramatic in vivo data for is this approach with doing local delivery of methylprednisolone. So we said, oh, this is very well, but look, methylprednisolone is being used in the clinic right now. It's being given in high doses systemically. Can we just improve that by using local delivery technology from the lab? And we had a postdoc who was very interested in doing that, and we made some nanoparticles to do this. Again, this is a glucocorticoid, methylprednisolone, and there have been two large clinical studies, or actually a third one underway, all showing some benefit with methylprednisolone systemic therapy, but lots of problems. You need a very, very large intravenous dosage. You give 30 milligrams per kilo dosage after, and you get infection, pneumonia, septic shock, delayed wound healing, all the problems you would get when giving high-dose steroid therapy. And there's a time window where you have to go and do this. This makes sense because the early inflammation is what you're targeting, so you need to do this quickly. So that's what's done now. So what we did was we made some PLGA particles, and I know there are some here that also play with PLGA. There's nothing fancy. We made some double emulsion particles of PLGA, and these particles are between 200 and 800 nanometers. We didn't worry too much about trying to get a type of control. And what we did was we made a hemisection to the spinal cord. And we said, okay, let's make an injury like this. Let's not treat it. Treat it with saline film particles that slowly release saline or methylprednisolone over time, excuse me. Or do what's clinically done. Give a 30 milligrams per kilo dosage of systemic methylprednisolone, which is because this is what's done clinically. Or give methylprednisolone that has been encapsulated in particles that slowly degrade and release methylprednisolone. Now, when I say slowly degrade, given that you're targeting early inflammation, given that you know the scar stabilizes over 2 weeks, I think if you do the first 3 or 4 days, I think that's the early events that you're targeting with steroid therapy. The other thing you should know about methylprednisolone therapy, as I said in the beginning of my talk, has nothing to do with regeneration. It's really trying to spare the death of more neurons. So you're trying to stop more neurons from dying, or more axons from damage. You're not really trying to regenerate nerves that are already cut or damaged. Okay, so this is just limiting the extent of injury. So if you do that, if you make a hemisection and put in nanoparticles into the spinal cord, and we do this just by injecting nanoparticles and mix them with a little thermoreversible gel to keep them there. So if you do that, I'm sorry, so I had this slide I forgot. So we did characterize the release of methylprednisolone in vitro. We were just targeting the first 96 hours to get a big burst. We don't mind the big burst in this case. We showed that the released methylprednisolone is actually active by looking at nitric oxide production from activated macrophages. You can take macrophages, activate them with LPS, lipopolysaccharide, and see that nitric oxide production is a standard assay for macrophage activity, and show that you can inhibit that by the released MP. So we characterize the particles and we try it in vivo. Now this is a representative section of a spinal cord that has been treated with methylprednisolone locally or not really. Now on the left here is when we treated with just the saline film nanoparticles. The initial cut, just for your calculation, is no deeper than about yay much. So all this extra growth that you get is secondary injury in terms of growth. Again, the colors mean something. So the GFAP is the reactive astrocytes, and that's in red, and the neurofilament 160 are the processes and neurons that are expected. So all the green stuff is neurostuff, all the red stuff is reactive glia. We also have very similar pictures. If you look at chondroitin sulfate proteoglycans, they're co-expressed with all the red stuff. So anyway, upon at least our impression initially is that there's a lot smaller cavity here than here. Okay, but really you have to look at it more closely. And I'll show you some movies. Hopefully these work. So if you do a movie of a bat walking across a beam, an intact animal has no problem walking across. Okay. 2 weeks afterwards, if you injure the animal, do a T9 hemisection, and injure the animal, you can see the hind limbs it can't use. It can drag. So, but it can use its forelimbs, it can drink water on its own, so it basically walks across, it drags its foot. If you treat the animal with methylprednisolone, notice the hind limbs, notice the weight bearing, they walk across really well. So the actual quantitative data is here. When you count the number of times this hind limb slips off the beam, and that's what these numbers are. And again, the lower the better. Now, an intact animal has no problem, as I told you, walking across. So this number is really low. It really doesn't slip. If you treat it with methylprednisolone, it's over here. That's the methylprednisolone with nanoparticles. This is systemic methylprednisolone. This is where we are clinically right now. Better than this, but really not very good, because you can imagine, because systemic methylprednisolone, not much gets to the spinal cord problem. And this is if you do nothing. Now we have data showing this seems to be coming down spontaneously. We have data that this levels off at 6 weeks. That this doesn't get any worse at 6 weeks. So it's not like this spontaneously going, this is real. And we see significantly better results with local delivery of an existing known therapy, methylprednisolone, nothing fancy. And the dosage of methylprednisolone, the total methylprednisolone that we give in these animals is 1/300th of what you give systemically. Okay, so you're rescuing basically We have another test with intact animals walking across a grid. Again, the animal can sense the grid, doesn't slip really. You can notice the hind limbs. It does a pretty good job walking across the grid. If you injure the animal, treat it with, again, saline, you can notice the slips. The hind limbs just keep slipping. And you can count the number of times it slips in a unit time. There you go. So it doesn't do very well with the hind limbs. You can see the hind limbs slipping again. Again, when you treat with local nanoparticles with methylprednisolone, they do much better. They don't slip. They don't slip much at all. So again, you can quantify this. Again, lower number is better. Again, normal animals have no problem. They never slip. When you treat it with methylprednisolone, very little slipping. That's why this movie would have to run a long time to show you some slipping. Again, systemic is in the middle, and no treatment does this. Again, this also levels off. It doesn't keep improving. Again, this is, I guess, where the rubber meets the road, as people say. So behaviorally, we see big differences, and we've quantified the lesion areas. I have detailed data of this. We just submitted a large BRT on some of this work, showing that you do alleviate cell death and rescue neurons a lot more when you do local delivery. But so the nanoparticle mediated delivery significantly decreases secondary injury. We're interested in nanoparticles in this application because we want really, really small signatures of any polymer we put in the nerve gap in the spinal cord. Because we want nerves to grow through, we don't want them to be obstructed. But the more significant result in my mind is this thing. So we use 100 microgram per kilo. This is 300 times lower methylprednisolone given. Locally, I have not looked at systemic side effects and such and that, but it's logical to think that they would be low. So I want to conclude this presentation pretty much by saying that understanding the mechanism of inhibition in the CNS will give us, I think, better, more focused and more targeted ways to try to design therapies to overcome that inhibition. And I think this is important and it's missing information that we need to generate in our case. And tissue engineers are not, I know this is the applied day, I think we still do fundamental research and use tissue engineering models to develop better understanding. So that's what I mean here. Spatio-temporally controlled delivery, because it's local, it's temporally controlled. We've also made nanoparticles to deliver BDNF and a whole bunch of other things. We have an approach in this BRP to get more efficient delivery and testing. And we probably need, in spinal cord injury, we probably need anti-inflammatory therapy, something in the lesion to get them to grow, something against scar to make them grow through scar, and maybe something even distal to attract them to come over. My feeling honestly is that to get regeneration in the spinal cord, you need a multifactorial approach. And most labs do one or the other. The technology to do this simultaneously doesn't exist. And I think our group and perhaps our collaborators will attempt to do that. Rather than discovering new drugs and things which we always keep an eye out for, I think if we do this in a coordinated fashion, controlling in space and time the therapy, using scaffolds and delivery vehicles and where we put them in different factors, I think we have some chance, hopefully in the future, of doing this. I'd like to thank Ryan Gilbert, who did most of the CS gag work. He's a professor now in Michigan. Anjana, a graduate student who did the Road GTPS work. Zhao Jin, who did some of the early work with this, is a professor at Stevens. Bob McEwen, my collaborator at Emory, a postdoc, another postdoc, and Swami. Thank you for your attention.
Let's take 2 minutes for questions. Any questions out there?
My question concerning the
Wait for the microphone, please.
my question concerning the main inhibition mechanism of the regeneration of the gross and the central nervous system. I think it's already believed since a long time that the main inhibition mechanisms well established is like many of the growth factors are turned off after the development of the central nervous system. So it's very beautiful result that you've shown here. Just you focus on the role of sugar. That's right. Have you investigated the role of the growth
you ask a good question. There is lots of data showing that the trophic factors are not present as much as the PNS. The reason we didn't do that is because we know we can deliver them, and we know others have delivered them, and that doesn't help. In the sense it helps, but it doesn't cross the sky, right? So we do this work in the context of defining the problem as we have many approaches, I believe, already, including cell transplantation, to get, if you have a little gap in the spinal cord, to get things to grow, but we have nothing to cross the distal sky. They always stop at the distal interface. Trophic factors are certainly an option. In our combinatorial approach, we will certainly include trophic factors in doing that. Yeah. But I do not believe we currently have a way of making these growth cones cross the sky. And that's where...
have problems even for growth factors, even
Yeah, so if you take growth factors and you upregulate them, no matter where,
cross.
yeah,
Oh,
they
okay.
do not cross. We need to do something about sky.
Over here is a question.
I would like to ask, just for my interest, how long of time did it take this research to study the difference in chemical composition between scar in the brain and normal cortex, how to inhibit it? How did it take long of time, this research, until you come to this result?
Depends on the postdoc I've had, the quality of the student. No, I'm not sure I exactly understand. So if the time frame of the experiments are such that in vitro experiments, we typically do them for 4 days or 6 days because we get enough growth. The in vivo experiments, our 6-week is in infant time before we look at scar, 2 weeks, 4 weeks, 6 weeks. The data, it took 5 years for the student to get a PhD, but I guess it's any project, it's funny because you do a lot of groundwork and the actual experiments don't take that long. So it took us about 1.5 years to 2 years to do all those experiments.
2 years?
To do the experiments, but to know how to do
The
them.
data you said about 5 years. Or?
Yeah, so it's hard to because things happen in parallel. But,
No, I think even if it's 5 years, it's very short time in comparison to you.
yeah, well,
At least you achieve something, I think.
yeah, well, thank you.
Thank you.
Yes.
Let's do Rania, and then we're going to move to the back of the room.
My question is, how do you deliver the nanoparticles to the site of injury? And once you deliver them, are you sure that they stay there and don't go anywhere else?
Very good. So the question was, how do you deliver them? So they're so small that you can inject them. And we actually, to make sure they're there, we mix them with this agarose gel I talked to you about earlier. We have a paper that came out, I can give you a reference, showing how we do that. That was published in Biomaterials this year. They have a special issue on neurodegeneration. So we use the gel to keep them there. We've done experiments putting dyes into the particles to track them and to see if they're eaten up by cells or what. The conclusive results we don't have yet. Okay. But we've looked 3 days later, 4 days later, 7 days later. We see the dye, but we do not have conclusive results if they're inside macrophages, for example. So I don't know. If we do find from those experiments, which we'll know in a few weeks, that they're being eaten up. In this case, it didn't matter with methylprednisolone because those were the target cells, right? So I don't mind if they're eaten up. But if I want to release trophic factors, then I wouldn't want that. If that's the case, I think if we coat them with PEG or something like this, we'll decrease the uptake by those cells. We have a couple of ideas on how to do that, if we find that to be a problem. But I think we have ways to get rid of it.
Okay. Back.
Yes, sir.
Yes. Actually, a lot of people think that embryonic stem cell therapy is the future, and you have elaborated on 3 strategies not including this. Can you elaborate a little bit on the current status of this kind of therapy?
Yeah. So I've always been skeptical about stem cells and CNS repair or PNS repair because anatomically there's no way to transplant cells in the middle of a pathway to recreate the pathway. All the results that people are reporting, and people are beginning to report positive results with stem cell therapy, is that they turn into glial cells. The stem cells differentiate most of the times in vivo into glia. And they help, there is some evidence from John McDonald's group and others do it, showing that they myelinate the nerves better. So the nerves that are demyelinated and therefore not so functional Glia also make trophic factors and such. So the transplanted cells are functioning as support cells when you transplant them. So that is certainly a viable approach, but it is not going to regenerate a severed fiber, except in the fact that it may act as a trophic factor producer and things like this. So, but they're not going to become a participant in the circuit, I do not think, because that's tough. So I don't know if that addresses the question.
Let's take one more question, and then I think we should take the break, come back to the general discussion.
Well, actually, I don't have much background about your subject, but why didn't you try simply to bypass the scar?
Sorry? To bypass the scar?
Yes, you're having a problem with bypassing it, so use on the magnitude that you mentioned in the first lecture.
So you can, so if there is so you'd have to, when you try to bypass the scar, you'll have to enter the scar someplace. Anything you do to enter the scar, you will make scar, right? Enter the card at any time, you will make scar. So people have tried, Mary's Bunge's group, to deliver chondritis ABC chronically. It turns out that the chondritis ABC is also thermally not stable. It degrades at 37 degrees within an hour. So to get around that, people put in these mini Alzheimer's pump type things in the car to slowly drip the enzyme. So people study chondritis ABC. And when they put those very fine things, that they make scar in spondyl again. So at some point you'll have to enter this car. Whenever you do, you make